You have came across multiple terms such as mitotic apparatus, centrosome, asters, astral rays while reading about cell cycle and cell division. I'm sure you must be having a lot of queries related to these. So today we are going to discuss in detail about the mitotic apparatus. So before going into the detail of mitotic apparatus, let's talk about centrioles. So centrioles are composed of microtubule triplets, which are 9 in number, and these microtubule triplets are connected to each other with the help of C2A connective fibers. And further, they are connected to a central rod, also called as hub, with the help of 9 spokes. This particular arrangement is called as 9+0 arrangement, and because of this arrangement, centriole gets their characteristic cartwheel appearance. Now the question comes into our mind is that where we can find these centrioles. So we can find these centrioles in animals, few of the lower plants, protists and fungi. In high plants, we are not going to see these organelles. Now, the next important question is that what is their importance? So, they basically constitute the mitotic apparatus. Also, one important point is that their duplication takes place in S phase. That means a mother centriole duplicates itself to form a daughter centriole, and together they are going to constitute the next structure, which is called as centrosome. So, what is centrosome? Centrosome is when two centrioles, that means the mother centriole and the daughter centriole, align perpendicular to each other. Now, because of this perpendicular alignment, they get their characteristic T shape, right? Now, let's move ahead to understand about the mitotic apparatus. Now, first structure that we need to understand is aster. So, what is aster? So, we have learned that centrosome duplication takes place in interphase, then it moves towards the opposite pole and radiates out the microtubules. So, one centrosome, along with these radiating microtubules, constitutes the aster. Right? So this particular structure is aster. The microtubules that are being radiated out of the centrosome, these microtubules are called as astral microtubules or astral rays. So what we have learned from here is that one aster is composed of centrosome along with the astral rays. That means the microtubules that are radiating out of it at the polar region of the cell. What is a mitotic apparatus? It constitutes of two aster along with the spindle fibers. These spindle fibers can either be of polar nature, polar microtubules, or kinetochore microtubules. Now let's see the structure of mitotic apparatus. So when we talk about the mitotic apparatus, the centrosomes start, radiating out the microtubules from them to align the chromosomes at the metaphasic plate. So these microtubules can be either astral microtubules or astral rays that are present at the polar region, could be kinetochore microtubules which are connecting the polar region to the kinetochore region of the chromosome, or polar microtubules which are basically stabilizing the mitotic apparatus, and that is how the entire mitotic apparatus will be formed from two asters at two different poles and the associated spindle fibers. So that's all for the mitotic apparatus. Hope you understood it well. Thank you very much.